Hi everyone. So far we have done dimorphic fungus, and the goal for the today is to get done with as many 2024 microbiology topics as we can. Ignore the lighting for the day. So yeah, let's start with it. So the first topic of the day is leptospirosis. Now, leptospirosis is mainly causing your icterohemorrhagic fever. So what do we mean by icterohemorrhagic fever? You can see here. First things first, ictero, that means you have jaundice, and then you have hemorrhagic fever. So you have some sort of hemorrhagic manifestation. You can have hematuria, or you can have hemoptysis. Any sort of hemorrhagic manifestation counts here. So icterohemorrhagic fever, and obviously you will have fever. So remember, in what is leptospirosis? So leptospirosis is a type of spirochetes. So I'll just write it here. So it is a type of spirochetes. Okay, yeah. Now, icterohemorrhagic fever, we have already discussed that, and it is a NEET 2024 PYQ. You can clearly see that. And in NEET 2024, they have given a clinical scenario that the patient might be having some sort of jaundice and some sort of hemorrhagic fever. How does it spread? So it spreads via contaminated water and mostly via rat urine and rice field worker. So probably you will have a rice field worker or farmer in the clinical stem. So yeah, that's that. And most important in leptospirosis is your agglutination reaction. So we have mainly three types of agglutination reaction, which is CAT, SAT, MAT, which is cold agglutination reaction. Then you have standard agglutination reaction, and then you have microscopic agglutination reaction, right? So my Billy, so this is the mnemonic which one of my friend told me. I think she has learned from Preeti ma'am, I don't know, but one of my friend suggested me this mnemonic while I was preparing for my PG. So this is the great mnemonic, which is CAT, SAT, MAT, my Billy. So cold agglutination test will be having for the mycoplasma. For standard agglutination test, you have brucellosis, and for microscopic agglutination, which is the disease that we are talking, is leptospirosis. Leptospirosis, again, we have already discussed that it is a type of spirochetes, and it has got this hooked ends, you can see. Okay, so that sorts your leptospirosis. I feel leptospirosis is the current favorite topic of examiner. Not each and every year, every alternate year, they just keep asking leptospirosis. So yeah, that's that. The next thing is Salmonella typhi. Now, Salmonella typhi, we all know what does it cause. It causes typhoid, right? I'll just write it down. So it causes typhoid. Now, so the one thing which is a hot favorite question of every examiner in Viva and in clinical STEM also you will find. So naturally what happens is whenever we have fever, our body has tachycardia. So for 1 degree Celsius, we have 10 beats which goes up. So if 1 degree higher fever, your 10 heart rate, 10 beats per minute will increase. So it's 10 beats per minute. But what happens in typhoid is, despite you having very high fever, you will have bradycardia, which is classically known as Fagit's sign. So this is the question which I got asked in Viva, and in typhoid clinical scenario, they mentioned this, that the patient is having fever, but still the patient's heart rate is low. So that means you have bradycardia. So that's that. And the question which is asked from typhoid is this, which is on week 1, what do you do? In week 2, what do you do? Week 3, what do you do? And week 4. So this is not my mnemonic. 100% credit goes to my faculties in second year. They taught me this mnemonic. So yeah, I think in the book which we referred also had this mnemonic. So yeah, BASU, which is blood in week 1. Then you have antigen antibody reaction, which is your Vidal test in week 2. And then you have week 3 stool culture, and week 4 is urine culture. Again, very important thing you need to know. So Fagit sign is done. What happens in Fagit sign I have already explained. Then you have also spots, which is your red spot all over your body, predominantly over the back. So that is known as Roth's spot. And you also have this jet black colonies. So jet black colonies over Wilson Blair medium, right? Jet black colonies over Wilson Blair. Wilson Blair medium is very characteristic for Salmonella typhi. And Salmonella typhi is of course gram-negative bacteria. We all know that. Okay. So what do they actually ask in Salmonella typhi? Again, clinical STEM question. Now, one thing which I want to mention is in Salmonella typhi, you have got O antigen and then you have H antigen. So in NEET 2024, they also asked that what is H antigen? So O antigen is somatic antigen, but H antigen is your flagellar antigen. Okay. Yeah. So again, what is O antigen? O antigen is somatic antigen, H antigen is flagellar antigen, and which is more immunogenic. So remember this, that H antigen is more immunogenic, right? And O antigen is somatic antigen. So it is same for S typhi, S paratyphi A, S paratyphi B. But in contrast, H antigen is different for each and every species. So it is different for S typhi, then you have it is different for S paratyphi A, it is different for S paratyphi B. So that's that. I'll erase this. So yeah. The next is parvovirus. Oops, it's not. Just sorry. So parvovirus. This is the parvovirus kit. So in parvovirus, so in NEET 2024, what was the question asked? So in NEET 2024, they asked that, a patient is of sickle cell and then they have aplastic crisis. So which virus causes aplastic crisis in sickle cell patient? So it is your parvovirus, right? Aplastic crisis in any patient of hemolytic anemia. Sickle cell was given in NEET 2024. Then it also causes pure red cell aplasia and hydrops fetalis. So we know in OBGYN we study about hydrops fetalis a lot. So we have two types of hydrops fetalis. One is immunogenic and one is non-immunogenic. So parvovirus causes non-immunogenic hydrops fetalis. What causes immunogenic? It is Rh negative pregnancy, and that's your predominantly obstetric causes. Here it is non-immunogenic hydrops fetalis, which is predominantly caused by parvovirus. Again, parvovirus is smallest DNA virus and it is the only remember, all DNA virus have got double-stranded DNA, but parvovirus is the only DNA virus which has got single-stranded DNA. It is very small DNA virus, and in body, all over the body, you have lacy white reticular pattern, and in face, you have macular erythematous rash. So that's your parvovirus. So again, leptospirosis, spirochetes, Salmonella typhi is your bacteria, which is gram-negative bacteria. Usually all the culture medias of Shigella and Salmonella typhi are same, but very characteristic of Salmonella typhi is your Wilson-Blair medium, which produces jet black colonies. Remember, jet black colonies. And again, so your week 1, week 2, week 3, week 4. Then you have parvovirus. Parvovirus, you just have to remember that it is very small, the smallest DNA virus, and it is single-stranded DNA virus again, and maculopapular rashes on the face and reticular white pattern all over the body, and the complication that parvovirus causes, which is your aplastic crisis, which is your pure red cell aplasia, and hydrops fetalis, which is again a non-immune type.